So it's a privilege to introduce Kit Curtius, who is joining us from San Diego, or the neighborhood of San Diego, California. Kit is a assistant professor at UC San Diego, and she basically has done a bachelor's in maths from UCLA. So she obviously she started in California, her academic career, moved on to Washington University to do a PhD at Fred Hutchinson with Georg Lubeck, and then move on to London at the Barrett's Institute, Cancer Institute, to work with Trevor Graham on groundbreaking research, some of which I'm sure we'll see listening to today. She is just starting her own group at UCSD as a new faculty in a group she calls Quantitative Cancer Control. So that's a very intriguing name. I hope that she can comment on that. She is one of the leading lights when it comes to cancer evolution, especially in the context of Barrett's esophagus. So without further ado, Kit, please take it.
Thank you so much, David. Nice introduction, and I'm so happy to be here. I wish I could be with you all in person. I've never been to Tampa, Florida, so I hope to go at some point. Yeah, coming in live here from San Diego. My name is Kit. I'm going to be talking to you about computational modeling of cancer evolution to optimize screening and surveillance. I've heard that this is sort of an interactive type of webinar, so if you do have questions throughout, say them loudly and I'll try to explain and go slowly so we can talk about the details. You saw a little bit of my path in just that introduction, but I thought I'd put it on a map so you know about my expertise. So I'm actually from here in San Diego, and yeah, went up the West Coast for my degrees. So mathematics and applied mathematics for my PhD, for those of you who don't know me. During that time, I worked a lot with a group in the NIH that was looking at Barrett's esophagus and esophageal adenocarcinoma and how we can increase prevention of this type of cancer through screening and surveillance. And some of the people that were involved with that will come up in this work. And as you heard, I moved to London in 2016. I joined Trevor Graham's group at Barrett's Cancer Institute and started studying another pre-malignant disease called inflammatory bowel disease that predisposes patients to colorectal cancer. So there was some parallels here between these types of GI disorders. And there I got a lot of expertise beyond mathematical modeling, but more hands-on with the genomics and bioinformatics work that we were doing in Trevor's lab. And David mentioned about work with Trevor in the introduction, but actually a lot of this work I'm talking about today started back in Seattle when I was working with people in the NIH, and is now kind of coming to fruition back in the States again. So you'll have to listen to another talk to hear more about the work with Trevor. But then just recently I moved back to San Diego. I was mentioning to David and Sandy that I actually haven't even seen my office yet. I started my group in July, but we are all computational, and so we are all remote still to stop the spread of coronavirus here in San Diego. So I want to sort of motivate this talk by talking about early detection. So what we really know from the data is that detecting cancer earlier saves more lives. So this diagram is from Cancer Research UK website, and it's showing the distribution of cancer stages diagnosed in 2014 in England. And as you can see, about 25% were diagnosed of all types of cancer in stage 1, and nearly as many were diagnosed in stage 4. But what you can see is that there's a stark difference in 10-year survival. So those diagnosed at stage 1 can live past 10 years, over 90% of patients, and then stage 4 has a very more dismal view of their sort of less than 10% survival by 10 years. And also the treatment costs are extremely different. So it costs nearly 4 times as much to treat early or late stage colorectal cancer versus early cancer. So there's a big push to try and diagnose more of these later stage patients into stage 1 because they have better survival and it's better on the healthcare system. So I like to think of this process of early detection as sort of catching cancer in the act. So while patients are living from birth, we'd like to know and identify who is at the highest risk of developing certain life-threatening cancers so that we can intervene and reduce and eliminate this risk appropriately. And so what screening looks like, this is just pulled again from the NHS website in the UK, is that we have a population of people who are living their life, and at some point we give them a test to see if they have any sort of worrying condition. And that will depend on what sort of modality that test is and at what age these patients are given a screen. And after they have some sort of outcome, we decide whether they should just keep on living their life with no further action, if they need some support about what to do next, or if they actually need some sort of treatment or intervention. And this is sort of the paradigm of what cancer screening does. And two of the questions that we'll talk about here is how are we doing this currently, and is it working very well? And also, just as a hypothetical, how many lives could we be saving if we were doing screening more adequately or better? And so one of the questions is screening trials in general have problems to be done well. And for many cancer types, the benefits and the harms of different screening tests and the intervals of which they should be implemented are just unknown. So randomized comparison of testing is really warranted, but it's not possible when most people in high income countries have already undergone screening or they've refused screening and they're just not comparable to non-screened populations. And so usually there's just a decision to make at screening at a certain age of a certain type, and then we just record the outcomes of that decision and see what the benefits were. But this paper out in Science kind of outlines what maybe a better strategy would be is to actually have randomized screening arms in the population in a certain country where individuals are put randomly into different arms where there could be different screening tests, there could be different starting ages, and then you follow them up long-term and see as you go if the outcomes are different and if one arm is more beneficial than another, and they're calling this learning screening programs, where then you would update and get rid of certain arms that aren't working well and go to the more beneficial arms. This type of thing is already being done in Scandinavian countries where they have a better system for tracking patients who have been undergoing screening tests. But there's still some open questions as to how to do this process, which is sort of an ideal situation of randomized screening trials. And those questions are simply, who should be screened? Should we put everyone into certain screening programs? Should just certain at-risk populations, certain genders undergo screening? And then secondly, at what age should they be screened? How do we make the decisions as to when to start screening people? And this is a really hot topic in screening now because we're seeing things like earlier onset colorectal cancers are going up. So should we be screening more young people? So it's a very prominent topic right now. So the outline of my talk is going to be a little bit of background about this process of field cancerization in the human body. So how clones become more cancer prone while patients age, and also how we take a sort of multi-scale cancer modeling approach to define these cancerization processes. Then the two parts of the talk are going to be the who of screening. So we're going to be talking about Barrett's esophagus as our sort of example and the progression to esophageal adenocarcinoma in these patients. And then the when of when is the optimal time to screen for Barrett's for at-risk populations, and a little summary. So I want to talk to you about field cancerization and why we think that this is a sort of hope for early detection research. And so we had this paper out that was a big review looking into the evidence for field cancerization, where on panel A, you see a sort of sporadic classic tumor that has malignant cells growing in sort of a field of normal surrounding cells. But the better we do at genome sequencing and different types of methodological research is that when you look for it, you actually see a lot of times these malignant cells are surrounded by not typically normal cells, but actually cells that are altered. They could carry some types of mutations. They could have phenotypes that are different from normal, yet they could still look morphologically normal under a microscope. And so that this sort of field of cells actually looks like the precursors to these tumors that are arising in certain tissues. And these could be caused by random mutations that have a selective advantage and grow out in a population of cells, or it could be caused by mutagen insults, such as UV exposure or carcinogen from cigarette smoke. Does everyone still hear me okay?
Yeah, it sounds good.
Okay, good. I can't see you, so I just want to check in. So the process of field cancerization has a lot to do with what we call clonal evolution in aging humans. So with this study that we did for this review, the more that we looked into different tissue types, the more we saw evidence of this type of somatic evolution happening since birth across the human body. And so starting with some pretty famous studies that you will probably be aware of in Science in 2015, 2018, started with some great deep sequencing done on normal autopsy patients from sun-exposed skin on eyelids. And what they found were many different types of subclones that had outgrown that were clonally related in patients that do not have cancer at all. And so it's surprising to find actually genes that are cancer-promoting genes were clonally expanding and seen in certain parts of the tissue in normal skin in the eyelid. This was also seen in the esophagus. So looking at patients that were healthy, ranging from ages 20 to 75, to the researchers' surprise, they found that although the mutation rate was much higher in normal sun-exposed skin due to the UV exposure, actually the types of clones that grow in the esophagus carried a lot of cancer-promoting gene mutations. So these are arranged by age, and if you think of the esophagus as laying flat on a surface, the circles correspond to different clones that are sizes of cell populations that have that mutation. The colors are different mutations, and the size of the circle is the amount of tissue that carried that clonal mutation. And so you can see that there's many, many diverse types of clones undergoing this sort of process of evolution in human esophagus. And you can also just plot this as sort of number of mutations by age and see this significant effect. Also in the colon, within our own group at BARRETT'S, this is examples of mitochondrial enzyme cytochrome C oxidase deficient clones, where these deficient patches show clonal expansions. If you look sort of en face, you can see these blue crypts are undergoing fission, so they're dividing and growing in the colon. And again, others have found that point mutations increase also with age in the colon. So we see this sort of process of clones that have these mutations that are growing, and they're undergoing all the processes you expect in evolution. So genetic drift, natural selection, and this is just getting to be a really big area of research, as we all know, looking at also just normal tissues and how clonal evolution is happening within those tissues. But of course, this is sort of new fancy data. Some of the oldest data we have in cancer research is just cases. So number of cases in a specified time period by age in the population has been data we've been collecting for a long time in the US. So this is data from the Surveillance Epidemiology and End Results Program, or SCRE. And it's showing by age the colorectal cancer incident cases across calendar years. So between 1975 to 1979, all the way up to 2005 to 2008. And you can see this very sort of indicative shape of this curve, which is telling you something actually about the process. And this is sort of was a birth of mathematical modeling in cancer research. And so going back to the 70s and 80s, a big discovery was made by Alfred Knudsen looking at retinoblastoma genes in patients who were developing cancers in the retina in children. And he teamed up with a mathematician named Suresh Molgavkar, who's a colleague of mine. And they looked at how can we look at these incidence curves, say in the lung, and actually derive what was the sort of mechanistic process that made these curves have the shape that they do. And so this is sort of going to be the level of multi-scale I'm talking about, all the way from population level incidence down to sort of cellular clonal expansions. And so the model that we can think about is a multi-stage process. So this is sort of the diagram of work that we do in the colon, where you can have sort of sporadic random mutations occurring since birth. And many of these will be what we call unsuccessful. So they will undergo selection where they'll be unselected in the population and go extinct because they harbor some mutations that aren't beneficial for the cell. And some initiations will start to grow out. And so the precursor to colorectal cancer is called an adenoma. And unfortunately, for some people, they will have adenomas that grow quite large that can undergo malignant transformation that will lead to eventually a symptomatic detection of a cancer. And so the way that we write down the end result, which is the symptomatic detections, is this incidence rate, which is number of new cases in a specified period over the number of years, person years at risk. And so the nice thing about mathematical models is we can actually describe this process quite formally. So the model in sort of a dynamic sense looks like this. So you can have initiation of cells with Poisson initiation rates for mutations that initiate pre-malignant clonal expansions. In the case of colon, it's going to be the adenoma. And they can have their own birth and death rates, alpha P and beta P, and they can undergo malignant mutation with rate mu2, for which small preclinical cancers can grow at their own growth rates, alpha M and beta M, and eventually may be detected at a certain sort of size-based detection parameter rho. So these are, this is a stochastic model. It's what we call a multi-type branching process. So the types are these sort of P cells and M cells, and it's a birth-death mutation process sort of linked together. So when you have a formal model like this, you can actually write down what this function is. So in biostatistics, this is called a hazard function. So it's the limit of the time of cancer occurring between T and delta T, given that cancer has not occurred yet over delta T. And when you write down actually this function, given this model of differential equations, you can pull out what these incidence curves actually reflect in the biological process. So this exponential power law behavior in the age incidence curves reflects the pre-malignant growth phase, so that alpha P minus beta P. And the slope has to do with the multiplication of the rates of initiation and the probability of survival for the pre-malignant clones, for example. So then when you actually take this hazard function from the model and fit it to incidence, you can recover these type of parameters like growth and mutation. So now we can talk about what are the expected rates of evolution given incidence. And we did this over many different types of cancers. This is back in 2013. So we applied this type of model in gastric, pancreatic, colorectal, and esophageal adenocarcinoma, and we're able to estimate biological parameters like mutation and growth rates from these cancer incidence curves. And of course, they're corrected for period and cohorts. They can be sort of standardized in that way. But you'll notice that esophageal adenocarcinoma looks a little different. First of all, it's seen a huge explosive amount of growth since 1975 in men. And what the curves, why they look a little bit different is actually, this is not the perfect model for esophageal adenocarcinoma. It actually needs an additional stage because you don't immediately go from normal to kind of pre-malignancies or what we call dysplasias in the esophagus. You actually go through a precursor phase called Barrett's esophagus. And so we include another transition in this model that has this rate nu of T that can depend on age. And that sort of gives this sort of characteristic turnover effect in the incidence curve. So I'm going to be focusing on this type of model for esophageal adenocarcinoma in particular. So the first part of this talk is about who are these Barrett's patients and why are they important in this process to esophageal adenocarcinoma. So for those of you that don't know, we'll do a little bit of background about the numbers that we talk about when we talk about Barrett's. So this is the view through an endoscope from an endoscopist looking down through the esophagus to the connection to the stomach. This is called the gastroesophageal junction. And normal tissue under the microscope can undergo this transformation to Barrett's esophagus, which is this salmon-colored mucosa that grows around the gastroesophageal junction and up the length of the esophagus. And the unfortunate things for patients with Barrett's is that they're at increased risk of developing esophageal endocarcinoma, or OAC here. And that typically goes through stages of changes in the phenotype of the cells through dysplastic stages. Unfortunately for the patients, they often don't know that they have this disease because it's asymptomatic. So it's sort of an adaptive response to mainly acid reflux or gastroesophageal reflux disease. And so we think that the prevalence in the population is about 1 to 2% from population studies, but oftentimes patients don't know they have it at all. And that's an important point. For patients with GERD, the literature says that they have up to a 5-fold increased risk of developing Barrett's esophagus versus a normal patient. And the reason we focus on these patients is that it's known that they have a 40 to 50-fold higher risk of esophageal adenocarcinoma than the general population. So that is extremely big difference in risk. But also at the same time, the rate of progression once you know you have Barrett's that doesn't have any dysplasia to cancer is very low. So only 0.3% of patients diagnosed with Barrett's will progress to esophageal adenocarcinoma each year. So this is a sort of numbers game that we have. And so because of that increased risk, this is how current approaches work. So in the US and the UK, screening is suggested for high-risk gastroesophageal reflux disease patients starting at age 50. And there's an extra list of risk factors such as being male, having obesity, et cetera. Those patients will typically be suggested to undergo an upper endoscopy. And then if you find Barrett's, which would look like this under the endoscope, you're suggested to go under lifelong surveillance to look for those dysplasias and early cancers, because we actually have very good treatment methods for removing those, typically with radiofrequency ablation. So how are we doing at that right now? This was the stark picture that we sort of looked at for a while when we started this project. We call it the Napoleongram because it's sort of like Napoleon's March. And what it shows is that in 2010, there's about 150 million people living in the US. And from population studies, we think about 20% of them had GERD, or about 30 million people. Of those 30 million people, it's expected that only about 10% of them will undergo an endoscopy. So that's already not very many of the total people that we think are the highest risk of Barrett's esophagus. But of those that do, we think about 8% of them were diagnosed with Barrett's, and that number will come up again. Unfortunately, once those patients are diagnosed and undergo lifelong surveillance, they only contribute 7% of the total 10,000 cases that we expect from SCRE data in a single calendar year. So actually, who are all these people? There's people who have GERD who never undergo endoscopy, and there's people without GERD who would never be suggested upper endoscopy based on the current guidelines now. So one question is, where are all these Barrett's patients when we're really only diagnosing a very small number of them? And so the very simple question that we aimed to answer in this first part of the talk was, does all this esophageal adenocarcinoma that you see in the SCRE data plausibly come from Barrett's esophagus patients? We have an idea of how much Barrett's we have, and we have an idea how much esophageal adenocarcinoma we have. Is there some sort of meeting up of these numbers? And the reason this is important is because if the answer is yes, then we should continue to focus on improving Barrett's detection because they're the right population to target. And if the answer is no, then we need to focus on identifying some sort of alternative pathways because Barrett's screening programs will have limited benefit. And I should mention that the reason that this question is so unknown and so controversial is because we haven't really had any examples of watching a normal patient progress straight to esophageal adenocarcinoma, just because they would never have been followed up that way. So there's a sort of big open question about this. And the reason we stepped in is because this is an easy question to answer on the back of an envelope. Because there's just all these combinations of calendar year and birth cohort and age, you really need a sort of quantitative model to answer this question. And this is where I feel like coming in as mathematicians who know a bit of biology, we really have a role to play in answering difficult questions like this for clinical impact.
Hey, Kit, it's Heiko. Can I ask a quick question?
Yes.
Hey, when you talk about the 50-fold increase in risk over the normal population if you have Barrett's, is this age dependent?
Yes, it is.
Does this number change in an older or younger population?
Yes, it is age dependent. So that's sort of like an overall age groups from those large population studies type of fold risk, but it's a really good point to make. So even, you know, of course, your risk isn't going to be that much increase if you're comparing 10-year-olds, for example, because there is that higher incidence later in life. So we'll get a little bit more into that sort of age specific. And I wrote that down actually to bring up that question of these are obviously not very useful numbers because they're kind of convoluting all these different ages. It's a really good point and it will be different. Okay. So of course we come in and we apply this model. So we've got this multi-stage clonal expansion model for esophageal adenocarcinoma. And I introduced that we've got this rate of Barrett's onset, which I call new, and how we incorporated the sort of dependence on these gastroesophageal reflux disease patients is by making this as sort of age dependent gastroesophageal reflux disease rate, where you've got the prevalence of GERD in the population at age T that has a relative risk RR, and then you've got the non-GERD population, 1 minus P of GERD. And this is the sort of baseline parameter that's pulled out from incidence data. And so we've done this actually back in 2014 with this model and fit to SCRE incidence. And the other stratification that you must do is using sex as a biological variable, because you see that the incidence between men and women is extremely different in this cancer. So there's many more cases in men than there are in women, as you see in these axes. And also it's important to separate by birth cohort, because there's different rates based on birth cohort, which are shown in the different colored lines. So the SCRE data is shown in the dashed lines, and the model fits are shown in the solid lines. And so it was able to kind of pull out these sort of high level parameters for the cell mutation and growth rates fit to SCRE. And that's—
Kit, sorry, why is that such a big difference between men and women?
It's a really big question. They don't know, but they really think it's something biological, that the rates of these sort of initiations and growth is different between men and women. There's some sort of sex-based difference there, but it's not exactly known.
Are your parameters different for men and women then?
Yes.
And what specific parameters are changing there?
So the biggest difference actually is in this pre-malignant growth phase. So the clones grow much slower in women, is what we learned from the incidence curves.
And so that fit is reflected, that dynamic emerges from that?
Yes.
Nice.
So
Okay, thanks.
What's known about environmental factors, and to what extent they play a role potentially for the difference between men and women? So to my understanding, for example, drinking kind of strong alcohols increases the odds of getting Barrett's esophagus. So what is known about this?
The role of risk factors like alcohol, they've been sort of tenuously important. They're sort of minimized by the overall effect of GERD being much more of a risk factor. The differences between drinking between men and women might have something to do with it, but I think the overall effect, I forget the odds ratio, is not huge for alcohol in developing Barrett's. I think obesity is a bigger trend, which again, those prevalences between men and women are slightly different in the population as well. So I think that there's a few different types of environmental things.
Does it look into types of alcohol? Because I mean, drinking beer or wine is one thing. Another thing is like Scotch or hard liquors that can be more damaging to esophagus and more prone to increase inflammation.
Yeah, it's a good point. I can't think of any studies off the top of my head that split up alcohol type looking at risk of Barrett's, but I'll look into it. So basically,
But Kit, on that same point, obviously there was a whole big thing about, you know, a glass of red wine a day or whatever is good for you, but that was to do with heart health, I think.
Yeah.
But is this potentially saying, but it's not good for your gut?
Yeah, I think that's the problem, is that those type of studies sort of focusing on one thing will, have a negative effect on something else. So I don't think what we know would suggest drinking a lot of wine for your esophagus health and stomach. Okay, great questions. Thank you so much. So basically, this is to show you that the input for the model is incidence, so EAC incidence, and gastroesophageal reflux disease prevalence. So now once we have the model, we can look at the intermediary stages. So what we looked at, be surprised, we saw a really close fit of the model prediction for BE prevalence in the general population, stratified by, of course, age and gender. And then in the GERD specific population, it gets a little more complicated because you've got this difference in relative risk for patients with GERD. So what you see in the model is the blue is men and the red is women. And the CORE data is shown again in the dashed lines. And so the solid lines are for RR equals 5. And then in the literature, the basic range that you find of the relative risk in GERD populations is between 2 and 6. And so this can be a sort of idea of what the range of GERD patients are, because they're going to have different onset ages. And so you expect that the total kind of GERD specific BE prevalence will be within sort of this expected range. And so that was sort of also fitting with what we found from the model to fit with CORE. But this is an area that I'll mention because I'm talking to a sort of mathematical audience, that the methods for disentangling the GERD population and non-GERD population is a type of modeling that we're still working on. So this is a sort of coarse-grained view of what we expect the sort of range of prevalence to be. But it still seemed to sort of correspond with what Corey found for GERD-specific populations BE prevalence. And I should say that this study that we compared to was the one that sort of inspired this 50+ screening guideline, because they basically looked at this line and they said it sort of gets largest at 50, and so we should just do that. So it was a very sort of ad hoc way of deciding this guideline in the first place. And this 8%, again, is what was said in that Nature review, should be the sort of expected prevalence of BE within GERD. So it's still sort of fitting in with what we expect the population to be. So the overall question is, how many cancers does the model predict in 2010? So the model predicted that 2.2 million adults had prevalent Barrett's in 2010, which is around 1.6% of the general US population over age 40. And so again, that sort of 1 to 2% prevalence is sort of what the model recovers. It seems to be in line with what we expect. And then we can compute with the model the expected number of EAC cases for men and women with the hazard function. So this lambda is the cumulative number of cases. This PY is the number of person years at risk, which you can get from the census data. And this HEAC is the hazard function that you can write out from the model as an equation with age I and birth cohort K, where CJ is the calendar year. And so when you actually compute this with the model for age groups greater than 40 in 2010, we get an overall sum of the male cases and the female cases to be 9,970 with these confidence intervals. And so that seems to be extremely close to the 10,000 that we were expecting from that sort of Napoleonogram that we looked at. And the last thing to check was then, what is the progression rate between Barrett's to cancer given this model? And that was between 0.1 and 0.2% for men. Again, very low progression, but you still recapitulate this number of cases that you expect. And so when you just take the SCRE data raw and not just look at the Nature review, we computed this analogous estimate and got 9,400 new cases from the SCRE data directly in 2010. So this seemed to be that the model then predicts the total number of cases to be exactly what we expect. So the answer to our question was yes. This means that the progression from BE to EAC is consistent with the total number of EAC cases reported, so that the Barrett's precursor must be responsible for the majority of EACs diagnosed in the population. And so this was a result that we just had accepted. You can look at the preprint on that. And of course, the next question is, so we know we're supposed to be targeting these Barrett's patients, but when should we recommend that they come in for screening? And so at the moment, this is a little diagram to show certain patients that would be suggested to be screened at age 50. There's going to be a wide variety among them of certain types of risks. Some people should come back at a certain designated time in the future. Some people should probably never come back because they're sort of this very low risk. And so there's this risk stratification problem of how do we know who those 0.3% are every year who are going to progress versus those that should not undergo lifelong surveillance at all. So that's sort of the next question. But this initial age, when to begin, is, again, it's unknown. So we're going to talk about that from the modeling perspective. This is some earlier work actually I shared at SMB a couple of years ago, but now it's come more into its own light and now has a preprint. So the idea here is that we're looking for these sort of windows of opportunity. And so what I mean is when patients age since birth, they'll have normal esophagus. And at some point, they're going to develop Barrett's if they ever will. And there's some distribution of when that occurs during a human lifespan versus if they go on to later stages, such as small dysplasias and later cancers. What we want to do is find a time to screen them when they have this sort of precursor condition, but it's not too late that they have this cancer. And I should mention that the reason why it's so important to protect from getting to later stage is because this is a very deadly cancer. So most patients who are found with late stage esophageal adenocarcinoma will die within the first year of diagnosis. So we really want to protect from this happening later in life with our screening exam, but try and get the biggest yield or the biggest bang for our buck of the screen initially. And then once you know someone has Barrett's, if you had that optimal time to screen them, T-star, our next decision would be when would be the optimal time to perform surveillance on them or ask them to come back for a follow-up. With the model, again, we can use sort of formal ways of describing this. So this is a probability that the precursor A has developed between before time TS. and also that cancer has not occurred by time TS. And so when we look at this from a modeling perspective, we got these certain sojourn times that we're trying to target. So we're trying to target these early onset Barrett's called TBE, and the screening to happen before some time of symptomatic cancer diagnosis would be the best time. And then surveillance happening maybe before another symptomatic cancer occurs. And so the goal really is to maximize this sort of objective function, if you use it in sort of optimal control theory notation, or a utility function. And again, the model sort of captures these sort of lineages of cells that are undergoing mutation that can undergo clonal expansion within crypts, and eventually some might lead to an incidental cancer. And when you have this formal model, again, we can actually write down analytically what this function is. So typically, I should mention that previous models that have been used to decide screening for USPSTF and guidelines in the US have used sort of microsimulation of Markov models, where they have large in silico populations that they simulate. They test 2 or 3 starting ages, and then they decide which one is best. But the nice thing about using sort of a more formal mathematical model is you can sort of look at all ages and just maximize with a single computation. So that was sort of the plus of doing it this way, rather than a model that microsimulates and can have lots of sensitivities. So I'm going to tell you about the notation and just the main results I'm going to share with you today, which is about initial screening. So initial screening is, again, maximizing this objective function, which is going to be computing TS star, the argmax of this function. So we can pull out the probability also that death has not occurred by time TS, which is an important point. So we don't want to screen too late that it's likely that the population would have died of other causes, so that we can get from mortality data in the US. And then you can split up these probabilities of having BE and not having cancer. And I also introduced this parameter W, because this is a proper probability here, but for an optimization problem, you don't need to have a proper probability. You could actually try and extra weight this cancer event from not occurring by time TS by increasing this weight W. So it's sort of a flexible framework to meet the need of what you expect the tolerable risk to be in a population. The second thing we'll talk about is costs. So once you pick a screening age, there's going to come associated costs with it. And I'm going to just share one example, which is an example of the number of futile or unnecessary endoscopies you expect until some later age TF for all those overdiagnosed patients in an initial amount of X screens. So those are patients who, from a probability standpoint, are the ones who will never go on to get cancer. There's going to be a certain number of those that are overdiagnosed within the prevalent population of BE at time TS. And then we're going to have an initial number of screens. This number of exams per patient year is something that we've computed from previous modeling. And then the total number of years that this patient's going to be on surveillance until some later time TF. The third thing is rescreening. So the nice thing about probabilities is that you can condition. So if you condition on a screening result of not having Barrett's at time TS1, you can look for then the next probable time that you have Barrett's before cancer and before death. And again, it's just going to be the maximization of this objective function. And lastly, what if you were positive for BE at time TS1? It's that example that I gave of, if a patient is diagnosed, they might want to know, what's my future risk at a later age TST of getting cancer? That might be one of the first questions that a patient would ask. And that's something that we can quantify with the model as well, where if you don't find any detectable pre-malignancy or malignant cells on the time of screen, but you do find Barrett's, what's the future risk of cancer? And so these are all things that we can compute in a single computation with the model. So the first results on initial screening are shown here. So this is for, again, the strata are the general population of males, males with GERD, females, and females with GERD. And this is showing a plot of that screening objective function on the z-axis, the screening age on the x-axis, and this weighting parameter on the y-axis. And again, I just computed this for 0 to 10 to show that this optimal age, which is shown in the contour plots, the maximum of this is this red line, can differ if you decide you want to put more weight on protecting a population from getting cancer. And the sort of shapes here are showing the optimal time for that W=1 case, which again was our objective function with W=1 in the proper probability. And so these were the optimal ages that we computed. So for general male population, it was age 64, for GERDs males 58, for general female 69, and for GERD female 64. And I show you these results because the cancer risk associated with this age was very low across these populations. So this is again, assuming this W equals 1, so it's a proper probability. And this is the sort of amount of risk that you would expect to have sort of missed by screening at that late age, which is all under 0.4%. So the rest of the results I'm going to show you are as the example of W equals 1. And so again, looking back to our previous results, we kind of validated this sort of prevalence we expect from the model output in these plots. And the way I made these, I didn't show you in the previous paper, is that you can condition on not having cancer and not having died of other causes. And that's sort of the point of using clinical data, is you will always have that sort of conditioning. You will always only be looking at a population who is cancer-free and who has not died of other causes yet. And that's something that we can compute with the model and compare. And so that's what this is the function that I computed for this comparison. And I'll mention that a large prospective clinical randomized controlled clinical trial called BEST-3 just came out last month in Lancet. And what they found was in 1,750 patients screened for Barrett's with a very sensitive screening modality called cytosponge TFF3, that they found 7.3% Barrett's prevalence in the population that had 52% women. So actually, this is going to be some combination of these two. And when you compute the same from our model with 52% women, it's around 6.3. So actually, they found an even higher prevalence of Barrett's in their population than what the model suggests. Now, the cheeky thing is, once you have this conditional probability, we can also compute what we really want, which is the joint probability. We really want to know the best window when patients haven't had any of these. So for that sort of analogous situation for joint probabilities, you get this from the model output, which is, again, these optimal ages for cancer screening are going to be the maximum of these objective functions. And the nice thing is, if you consider, again, that sort of heterogeneous population of GERD patients, so they can range widely since birth of having relative risk 2 to 6, even if you looked at them separately, this sort of maximization is going to stay unchanged for GERD patients. So even though there's a heterogeneous population of GERD, actually the optimal age that you suggest does not change as the model predicts. Now to talk about cost. So I picked these optimal ages to screen, and how does that compare cost-wise to, say, standard care? And I plot this to try and sort of visualize what I mean. So the surveillance that undergoes all BE patients who are overdiagnosed or successfully diagnosed looks like this. So if you screen at age 50, this is sort of the percentage of overdiagnosis that you have that's expected who will never go on to cancer. And this is the amount that you expect if you screen at some optimal age 58, say for GERD men. And those will undergo surveillance for their lifetime, but are predicted to never go on to cancer. And so the difference that we're looking at is what's the difference between sort of this green box and this red box. And when you compute that cost function from the model, you get this. So there's 0.67 times the initial number of screens when you screen at age 50 versus 0.61 times the initial number of screens if you screen at age 58. What does that all mean? Well, the first thing we notice is that if you look at previous modeling studies, actually they found that if you screen at 50 or 60 with highly sensitive screening, both of those were found to be cost-effective in the cost-effectiveness analysis. So actually anything in that range still seems to be plausible from that cost-effective standpoint. And then from this example, let's put some numbers to this. So if you look at sort of census data, you can predict that about 400,000 men with GERD were born in 1950. And then if you compute this cost function, this means that about 24,000 less endoscopies are expected if you screen at age 58 versus if you screen at age 50. Now, an endoscopy costs around $745 US each. So that implies about a savings of $17.9 million for the lifetime of this birth cohort alone. And of course, if you then do this every year for different birth cohorts that are age 50 and 58 at different calendar years, you expect proportionally that this number will also increase the number of costs. So we expect this is actually going to make a big difference. And the reason that we did this type of cost function is because we really do think this lifelong surveillance, where every surveillance interval is not tailored at all to the Barrett's patient, it doesn't look at age, It just says that if you have dysplasia or not, you come back in 3 years, or you come back in 1 year, et cetera, is a lot of money being spent on patients who are overdiagnosed. They will never go on to get cancer. The third result is about rescreening. So what if we screen and we do not find Barrett's? So this is the conditional probability of not having Barrett's at time TS1. When's the best time to come back next? And so, as I mentioned, this really depends on when you first screen. And this is something that the model would predict. And so we can sort of adaptively design our rescreens or our surveillance based on these conditional probabilities of what you find at an initial exam or a surveillance exam. And what you see is that if you screen at age 50, you have a much more likely probability of finding Barrett's at the next time point than if you screened at the later age 58. And so this kind of met up with the data that we expect. So there's been recorded that B positivity on a rescreen is about 2.3% from the CORI database. And so that's about the same that we find in men here if you screen at age 50. But if you were able to screen at an optimal time, your rescreening probability would be about 1%. So it really depends that when you first screen as to how much rescreening yield you will have. And again, that yield will have a sexual diagnosis that's very minimal. So it's expected that many of the cases that are esophageal adenocarcinomas come from patients that develop Barrett's before age 58, so that it doesn't really seem like there's a lot of utility in rescreening patients if you already find a negative result at the first screen. That was the result that we came to with that. And lastly, we talk about risk. So if we condition on the fact that you do have Barrett's at time before time TS1, we know that the onset of Barrett's happened at some age before that initial screening age. If we find no premalignancy and no malignancy in those patients, what is their future risk at some later time TS2? And I just want to bring this to the point of your attention that right now there's no clinical way to actually measure what time that Barrett's occurred. We just see it or we don't see it. But from a mathematical perspective, a much more specific thing would be to actually know when this TB occurred. So some people might have had Barrett's for 20 years, some people might have had it for 30 years, but because it's asymptomatic, they never knew. And so mathematically, that makes the difference of changing really what happened most probabilistically in the amount of time that you've had Barrett's before your time of diagnosis, which means that more evolution could have been happening, more natural selection and things could be occurring that you had no idea about and might not even be morphologically seen yet under the microscope or under the endoscopy report. So more specifically, you'd have a more refined—
So I have a question here. It's Mehdi from Moffitt, Mehdi Damaghi. What do you mean by more evolution happening here?
So basically, just from a probability standpoint, if you've got stochastic rates of initiation, the longer you live, the more likely you'll have some sort of stochastic initiation of, say, pre-malignant cells. And that's just a product of sort of a stochastic model with age. But it is something that we see, say, in incidence curves where there's more likely cancer and, say, adenoma prevalence in later ages, likely because you've lived a little longer and you've had more chance for that type of thing to be developing in your body. So it's just from a probability standpoint of elapsed time is sort of a risk marker in itself because you're aging. And the nice thing about the modeling, again, is that if you actually knew that time, you could get a much more specific estimation of risk for a specific patient, rather than just knowing they were diagnosed at some age, at their chronological age. They might have differing onset ages, which means that they have different amounts of time that sort of evolution could have been happening within that metaplastic tissue. So if you actually compute this for different initial screening ages and different taus, you can see that in the future EAC risk up until age 85, it depends on when the first screening occurred, which again is not taken into account in surveillance strategies now, and also when that tau happened, so when the initiation of Barrett's happened. So for example, if you screened at age 50, that's these dashed lines here. The difference between in both sexes, if someone developed Barrett's at age 20, which is this purple line, versus if they develop Barrett's at age 40, there's a 2.4 predicted relative risk between those two patients. So quite a big difference where we're not capturing any of that in clinical data now because we just can't measure that onset age specifically from clinical notes. So yeah, this future EAC risk is largely variable and depends on this initial screening age and the specific patient-specific age of Barrett's onset. And some previous work that I'll just briefly mention that we're still working on, we've got new studies going now, is looking at how can we actually measure this? So can we use molecular clocks for Barrett's tissue and actually estimate when that onset age happened? And so I'll just briefly say how we do this is by looking at DNA methylation. So methylation is something that happens with age that connects to cytosine guanine points on the genome. Well, methyl groups are added by DNA-MT transferases. And what you can see is that in a Barrett's patient, if you take a normal part of their esophagus tissue and a Barrett's esophagus tissue at some diagnosis age, you can see that their methylation profiles are different and that there's a definite aging profile that's faster in the Barrett's tissue than in the normal tissue. So that you know when a patient is diagnosed what their chronological age is, and if you know something about the aging rate within Barrett's, you can actually use Bayesian modeling to infer when that onset age happened, or what I call most recent common methylation. So when they used to be the same methylation, and sort of get an estimate of what that is in a patient-specific way. And what we found a few years back is that this actually varies a lot between patients. So in a population, we had a cohort of 30 patients, and we looked at these sort of matched samples between normal and Barrett's tissues. And so their diagnosis, or the time of the tissue collection happened at these horizontal lines between different sexes. And the box plots show the posterior estimates from the Bayesian estimation inferring their onset ages. And so we found using this model that actually the onset ages varied a lot based on their methylation clocks. And so for example, for these two patients who had tissue collected at age 80, they actually had, one had Barrett's for about 20 years and one was expected to have Barrett's for more like 40 years. So this would actually probably imply differing amounts of risk in these two patients, because one has had this sort of pre-malignant tissue sitting around longer than someone who looks exactly the same chronologically. So just summarize now, coming closer to a close. So the main sort of story of this all is that evolution from normal to cancer cells requires time, and that screening offers this important opportunity to measure field cancerization in action. And I'll put a plug in again for my group that David mentioned, calling it the Quantitative Cancer Control Laboratory, where we're using quantitative models to understand efforts in prevention and screening. And the way we did this in the talk today was using these multi-type branching process models to use sort of standard epidemiological methods, so computing things like hazard functions, and combining that with what is happening under the hood in the tumor evolutionary process of population genetics to infer these growth and mutation rates. There's lots of modeling work to be done here for anyone who's interested, and there's new models that are incorporating more sort of allele-specific types of mutations, and a lot of growth to be done in this area of mathematics, I believe. And the main message of the first part of the talk was that using such a model that sort of cohesively analyzes sort of different types of population level data, we found that Barrett's esophagus population is the right precursor group to target to prevent essentially all esophageal adenocarcinomas. And those patients are just not being found right now. They're just very under-screened population, and a lot of people who have Barrett's have no idea that they ever had it. Lastly, this is the plug that the stochastic models really allow you this opportunity to derive the distribution of when you expect these sort of windows of opportunity to happen that can help you determine from sort of a pre-hoc kind of way when the optimal age to screen for premalignancies could be. This type of model can be applied in other types of cancer sites, which we're looking into in different gastrointestinal premalignancies. And we hope that things like cancer learning screening programs can sort of also have mathematical modelers involved who can at least give a educated guess as to when initial screening ages should be mandated that currently are just sort of decided by experts, and then the outcomes are sort of measured and compared, but maybe we can put a little more thought into how to first assign these initial screening ages together. And again, a plug for the lab. So this is a sort of research areas that we're kicking off this year. So the sort of discovery of mechanisms and evolutionary timescales is sort of the thing I'm most into right now. So this is connecting these cancer evolutionary processes, so different types of mutations that confer a sort of malignant potential in certain cells with this sort of epidemiologically led data that looks at sort of benchmarks of prevalence and incidence within a population. How do we connect these things? We really need good modeling and good data science techniques. I think we need new biostatistical methods that we'll be working on that can incorporate longitudinal data and different data types. It's going to need large amounts of patient data and hopefully a lot of interdisciplinary expertise. What this lab, I should say, is within this PhD program in the at UCSD called Bioinformatics and Systems Biology. And lastly, I didn't talk about it today, but something especially I work in an IBD and I'm really passionate about is these sort of clinical translational tools. So we know about this increased risk while we do our research, but we'd really like to be able to express this to patients, also to let clinicians know what is most expected from, say, clinical studies to be informative for their actions, for deciding surveillance for patients, for deciding treatment strategies, et cetera, and sort of personalize this way more than it is now, which is very standardized across patient groups. With that, I'd just like to thank a lot of people. So my initial PhD group at Fred Hutch, my advisor Georg Luback and Suresh Mulgavkar. William Grady is the PI of the lab that does the methylation work at Fred Hutch, and Bill Hazelton. My previous postdoc advisor that I did a short postdoc with was the one that we thought about this back of the envelope question of how many EAC cases come from Barrett's, John Inadomi, who's now at University of Utah. Amitabh Chak has also been influential in that research from Case Western. Anoop Dewanji is an old time math collaborator who's at ISI in Calcutta, who's helped a lot with the derivations of all these screening windows of opportunity. Joel Rubenstein is the connection for Corey, who's really great at framing these clinical problems. And I highly suggest if you're a trainee or if you're looking into getting into translational work is to find people that you really connect with from the clinical standpoint, because they have lots of data and they have lots of insight as to what's most important to model. And I also like to thank Trevor and the group at BARRETT'S, which is also a place where I've been working on these projects and loads of other things. We've really helped build the progress of this research. A lot of funding sources, my fellowship that I've just finished from MRC in particular, and happy to take more questions now. I really appreciate your questions during the talk. I put my email here too, if you're wanting to talk about something more in depth. I'll be taking PhD students next this year and next year, and also will be advertising for postdoc soon, so just keep a lookout. And I really appreciate you all listening to this and my first YouTube live stream. I feel very privileged, and thank you to the organizers.
Yeah, thanks, Kate. Amazing talk. Obviously, this is a good time for further questions. I don't know if anyone has any right away. I know that Russ was talking about something before, but I don't know if he has satisfied his curiosity with the rest of the talk.
I do, if nobody else is going to jump in. So beautiful presentation, Kit, and I'm really excited to see that part of the focus of your lab is going to be trying to integrate kind of more mathematical mechanistic models into this, which of course is one of those big leaps. The kind of epidemiologic world has often more viewed this from a kind of age-dependent perspective, which obviously it is. But one of the confounding factors there, of course, is you might be the same age, but you could have lived a very different life.
Yeah.
And so you aren't mutationally the same somehow.
Yeah.
And, that example you showed at the end kind of spoke to that, like, they were very different in terms of the amount of time they'd had potentially the disease.
Mm-hmm.
And so understanding a bit more mechanistically what went on that caused that difference, right, would be potentially highly insightful. But of course, the data you have is at that population level, and you've got to work with what you've got. So in terms of then trying to connect back to the sort of more mechanistic side, are you working with collaborators there to dig deeper into that will integrate into these more population level models?
Yeah, definitely. So I think it's a really good point to make that you can only sort of go as coarse-grained as sort of the data allows. So we know a lot about GERD, we know a lot about that sex difference, and we're able to make this sort of general population distribution based on those things. But of course, there's going to be more and more factors like diet, obesity, that can sort of stratify these patients even further, where if you had the data to look at say the sort of mutation rates in obese people, you'd be able to also look at those as sort of a separate population of people that would have, those rates of change should be different. And so I think, getting more data about that sort of mechanistic difference will be crucial to make these models impactful, because right now it's as high level as we can with clinical data. And
so a kind of obvious follow-up to that is, we have a kind of interest in this, is the role of the immune system and immune responses to these early diseases, right? Are they potentially facilitating in some cases versus, suppressive in others? And what could potentially differentiate those two populations? Is that part of what you're looking at or thinking of looking at?
Definitely thinking about looking at this. I would say even more so maybe in the IBD research, because the immune system in that is obviously a huge factor. And right now we're getting this sort of heterogeneous look of patients who have hot sort of premalignancy and cold, and we don't really know how to tease apart. But especially in terms of the IBD research, sort of something I'm interested in is combining sort of microbiome data of patients who also have biopsies taken, so you can match sort of what's going on sort of in an immune cell level and what's going on in genetically and how those things interact. I think it's a really good point, and it'll tease apart a probably really important part of this. In the Barrett's side, I hope to as well. And so we've applied for some money to do that kind of research collaboratively, looking at immune scores and sort of multiplex immunohistochemistry of Barrett's tissues. And I think we're going to get there, but getting the patients involved is hard because, again, so many Barrett's patients are not being diagnosed. So they're not part of large clinical trials.
Well, good luck with the new lab.
Thank you.
Exciting.
Hey, Kit, it's Heiko again. Thank you. That was very, very beautiful. A quick question. You're jumping from Barrett's to EAC. Are you aware of any other markers that could be used that, instead of at the binary, you have early cancer or not, some expression in maybe biopsy samples or so that you could use to see a bit more dynamics in between to more personalized screening intervals?
It's a really good point. And I'm sorry I didn't mention it, because I should have been talking a little bit about biomarkers too, because once you screen, of course, you don't want to just know chronological age. We want to know much more about these patients. And so in Nature Medicine this week, there was a study looking at Barrett's esophagus progression to esophageal adenocarcinoma, looking at the genetics. And what has been found in Barrett's is that copy number alterations are highly predictive of future cancer progression. And you can measure them with very cheap sequencing. Low pass sequencing under 1x depth. And so if you're able to do things like that, you can risk stratify much better and probably tailor surveillance intervals based on genomics. And I think an area of research I want to work on with the lab is how do we incorporate those variables into these sort of risk prediction models better? Because I think it's where we're going. I mean, that study was very convincing and illuminating the fact that we can do so much more if we look at genetic biomarkers.
Very cool. Looking forward to seeing that.
I have a question as well. And I don't know if it's going to be a silly or a crazy sort of question. So either I'm silly or crazy, or maybe both. But I was very intrigued by the field cancerization part of it. I have a vague idea of what it's about from your work and from Mark Rice's work at Duke. Do you know if there is any sort of poor law correlating the sort of prevalence of field cancerization with benign cancers, with aggressive cancers? Is this something that decreases in a sort of logarithmic scale as you go for more aggressive events?
It's a good question. I don't know exactly if anyone's done this in sort of a pan-cancer look. Do field cancerized cancers that show signs of field cancerization early, are they more aggressive or more deadly? I think in general, there's always going to be this lead time bias problem where if you can detect it early, it'll look like the survival is better because you've identified them early, versus when you always find late stage, it always looks like they did worse. And so this is a big question in Barrett's. There are studies out there that look for esophagectomies, looking for field cancerization around, and they can't find it. And they find that those actually died sooner than the ones that you could find it. So that sort of spoke to maybe there is a difference in if some fields kind of evolve slower than ones that happen quite fast. And teasing that apart, I think, is a really good point. And I just don't know from the epidemiological studies now, I think, if in general, these slow growing field cancerized things are less deadly.
Thanks, Kate. And any more questions? Well, if not, thank you very much once again. It was a great talk. I am getting on the chat window lots of people expressing how thankful they are for seeing your talk today. So
thanks so much.
Obviously, we are very excited to see how you guys do on San Diego as soon as things go back to some sort of normal.
Yeah, I can't wait. All right.
Okay. Well, thanks everybody, and see you for the next one in a couple of weeks.
Thank you.
Bye.
Bye-bye.
Thanks. Bye, Kit.
Bye.